Patrick, you're going to introduce Andrea, yes?
Sure. So this is Andrea. I work with her at the Broad Institute. She got her undergraduate degree in mathematics at Chicago and her PhD in bioinformatics, biostatistics at UNC. And so she does a lot of work on RNA models for RNA sequencing and other related, things in that field. But she's brilliant and she's friendly and she's great, and she's going to be a great resource to have here. She's going to be talking about the bio stuff underpinning a lot of the work we do. So she was telling me she didn't know kind of what level she should be teaching at, because I told her there's a huge variety. So if there are things that you're confused about, and you should stop her and ask questions. I'm sure everyone else in the room also has those same questions too. So go ahead.
Wonderful. Thank you for that introduction. As Patrick mentioned, my formal training is in the math side of things. I work on a lot of translational biology problems, so that's why I'm talking to you about the biology half of things today. But at some point, I may have to answer your questions with because chemistry. And my formal training is not in chemistry. So if at any point you want to stop me and ask questions, there's going to be a lot of breadth in these talks. We're going to talk about a lot of different topics. And so if you have questions at any point, I may defer your question and say we're going to talk about that in a later lecture, but we have time to talk about your questions as they come up, if I'm not already planning to talk about it later. So this first session is the Cell and Molecular Biology Primer. So this is the introduction to DNA, to genetics, and this I would think of today as sort of the elegant side of genetics and inheritance, and tomorrow will be the messier side of sort of this is the theoretical model, and tomorrow will be more of the things that happen in practice. Just a really quick outline of what we're going to talk about. We're going to talk about what a cell is really briefly. I don't think it's super important for the kinds of work that we do here. What DNA is. This central dogma of biology is something we're going to spend a long time on, and I think that that's really critical to understand where we're going. We'll talk a little bit about the structure of the human genome, how many chromosomes we have, how many bases we have, and what the variability in the genome looks like. We will talk about how cells replicate themselves, specifically how the DNA replicates, and then we'll talk about how our cells pass DNA to the next generation. And then we'll have a little bit of an introduction of how genomic variation comes to be in the population. So there's a huge diversity of life on Earth. All of these are animals, and animals are multicellular organisms. So animal cells are pretty small. They generally measure between 1 and 100 micrometers. And humans have an estimated 40 trillion cells. So we have a lot of cells. Obviously, humans have a lot of different organs. So all of these organs require different cells that do different things. And so I've heard of stem cell research, the idea that stem cells proliferate and they turn into all different kinds of cells in the body. And so we're going to be talking about single cells, but bear in mind that all cells are not going to be identical in morphology and appearance. So sort of structure of an animal cell. I'm sure most of us have seen this picture a long time ago in high school biology. We are going to primarily talk about the nucleus of the cell, which is this bit in the middle, and that's where the DNA lives. And then we will talk pretty tangentially, but it does come up a fair amount, about the small amount of RNA in the mitochondria, which are these guys. As you may remember from high school biology, they're responsible for creating energy in the cell, but they also have their own very small genome. And so all of the DNA taken together, that is the chromosomes that live in the nucleus and this little tiny bit of DNA that lives in the mitochondria, constitutes the genome. So what is DNA? I'm going to go just one step above the biochemistry sort of piece of it and say DNA is, you can think of DNA as sort of like Legos, that it's a bunch of interjoining pieces, and each of those pieces has two components. There's a sugar piece, and so DNA stands for deoxyribonucleic acid. So deoxyribose is the sugar piece, and the nucleic acid piece is here. There are 4 of these, as we all know already. So you have adenine, guanine, thymine, and cytosine. And it's those 4 bases that can be joined in any combination by this phosphate group. It's represented here in green. And that sort of, biochemists will refer to this as the phosphate backbone. So that's the piece linking them together, and then the bases are sort of hanging off the side. So this is represented as a single strand, just to show you there are 4 bases and they can exist in an order. But a single strand of DNA is not chemically stable. These bases generally want to form covalent bonds with the things around them. And so single-stranded DNA is highly unstable and almost never exists in animal biology. Viruses are a different story. So DNA generally exists as a double-stranded molecule. So the A always pairs with the T, the G always pairs with the C, and vice versa. And so we have the same information in duplicate, and there's this very predictable mirroring structure. These are directional, so the molecules are not symmetrical. And so we refer to one end of the DNA strand as the 5' end and one end as the 3' end. And you'll notice they go in opposite directions, right? So the 5' end of one strand is the 3' end of the other strand. And so this becomes important when we start talking about transcription and replication and a lot of the technologies that use this, because it does matter which direction we're reading. Okay, so if we zoomed out a whole bunch from the little diagram that we looked at, we have this sort of ladder structure. And because of the physical properties of this molecule, what ends up is this ladder ends up twisting. And this is the classic double helix shape that Watson and Crick got the Nobel Prize for, right? And so ultimately, when you see this picture, what we're actually seeing is just a twisted ladder. Okay, so what is a gene? Gene, in my opinion, gene is the single most commonly misused genetic word by the popular press and by lay people in general. So we're going to give a rigorous biological definition of a gene. So we already know that the sequence of the bases encodes the information. And the DNA in every cell in the organism should be the same. There's a little asterisk there because there are some very, very rare exceptions. But in general, that's a safe assumption. And so a gene is this biological unit of information with a specific structure. A gene is defined in modern biology as the sequence of DNA that encodes one peptide or one single protein. And so in order to understand what we mean when we talk about encoding a single protein, we need to understand the central dogma of biology, which I imagine most of us have also seen at some point. And so the central dogma is DNA, RNA, protein. This is the process by which the information in the DNA goes to protein, which are the movers and shakers of the biological world. So DNA, we talked about DNA already. It's identical in every cell, mostly. It's a double-stranded nucleic acid chain. It exists primarily in the nucleus of the cell. And it's very stable in this double-stranded form. We'll talk a little bit more about what that looks like a little later. And so transcription is the process by which we go from DNA to RNA. So this is the process where we write a message using the DNA to instruct the cell to do something. There are a few types of RNA. We're going to talk primarily about messenger RNA. The other two will come in later, but they're really not nearly as interesting. And so that's essentially a single-stranded copy of the DNA, and that also has 4 bases, but because it's molecularly a little bit different, thiamine gets replaced with uracil, which is a very similar molecule, but that's abbreviated U. So this is a picture of transcription. And so I think the most useful way to think about this is that the RNA polymerase is going to move like a zipper, unzipping the 2 strands of the DNA along the template strand, which, yeah, so it moves this way. This is the coding strand, so this is the information that matches the RNA, with the exception of T and U being substituted out. And this is the template strand, so this is the thing that we're going to use this is the complement of the thing that the RNA is going to say. And so the RNA polymerase opens just a small area of the DNA, and the RNA gets added. They're free RNA nucleotides, and those get added in sequence, and they have a phosphate backbone very similar to the DNA. And so what we end up with is this long single-stranded molecule of RNA. So that the complementing bases strategy, just as in DNA, applies to RNA. We talked about this already. Okay, so when RNA from a particular gene is present in a sample, the gene is said to be expressed. Expression level, so how much RNA, is super variable across time points, across cell types, across cell states, and probably across environmental conditions. The variability of expression is not well understood because we don't have human samples from every possible time point and every possible condition. So expression can be mediated by chromatin state. We're going to talk about exactly what that means, the sequence context, transcription factors, and some of this is unknown.
Can you talk more about that?
Sure.
4 bullet points and I know nothing about.
Yep. So sequence context, we can demonstrate correlations between certain motifs or certain recurring sequences and whether or not something is transcribed. Usually, I can draw this. So if you imagine that this is your DNA, this is where your RNA starts getting made. And then you say you have a gene here. Often this region, just upstream of the gene, contains certain motifs that are favorable for RNA polymerase and other things to bind. And that starts, again, that process is complicated and not always well understood. The chromatin state is what we're going to talk about immediately now. Yeah.
So usually when we talk about epistasis or something like that,
Yeah.
one gene controlling the expression of another.
Yeah.
So is that mRNA situation the same sort of expression? Is that the same?
Yeah, that is exactly the kind of expression that's referred to there. Yep. And so we will get through this, but there are proteins called transcription factors. Those are encoded by mRNA, and they'll get to be mature proteins eventually. And so often there will be a complex of things, so a bunch of things that complex together in this area, and that's what those motifs are. And they end up attracting the RNA polymerase to this site. And so when we talk about epistasis, when we talk about one gene affecting the expression of another gene, often what biologically we're implying, we don't always measure this, but one of the things that we imply could be happening is that one of the genes, say gene A, is encoding one of these proteins, and it has an effect on the expression of gene B in this way.
So should I think of it as that there's always an issue with expression of a certain gene, and when we talk about epistasis and we talk about assuming it away, or we talk about computational challenges that are associated with that, we're just abstracting away from all of that always happens?
This does not always happen.
Does not.
There are several mechanisms by which this happens. There could be a gene C that we haven't measured that is actually this component. And gene A actually has an effect on that one. So these are very complicated relationships. And often with the statistics, we're just measuring the things we can measure and making the inference we can make. And they are often indicative of something causal or indicative of something real and biological. But the exact mechanism is often not illustrated by the statistical sort of processes. So we will spend a lot of time talking about the sort of molecular complications that make this whole system work. Is that enough for now? Okay, sweet. So a single gene can encode several different transcripts, and this is a process known as splicing. And this was my first project in Ben Neal's lab, so this is a topic that I get very excited about. So a gene, in general, if you think about it, this black line is representing the DNA, and then the exons are these blue boxes. This is a fairly common sort of pictorial notation for genes. You would say exon 1 is this first blue box, exon 2 is the second, and so on. And these bits that aren't highlighted in blue are the introns. And so introns are the continuous segments of DNA. That is backwards. There is an error in the slides. I apologize. The exons are the pieces that get kept. The introns. So this should say introns are the continuous segments of sequence not preserved in the mature transcript.
So is that whole line one gene? Or
Yeah. So if you thought of this as the DNA and transcription start site was here, and I will add that in a second, and the gene is here, the exons are the bits we're going to keep, and I think I've animated this. We're going to come back to that in a second. Okay, this is a picture that shows what I'm talking about. So these pieces are highlighted in colors, and splicing is this idea that we're cutting out these little bits of line in between the boxes. And this splicing behavior can happen in a variety of different ways. So one gene, the same sequence, depending on the splicing machinery and the other factors present, can give you, this is called constitutive splicing, where you get all the exons in order. Exon skipping, we might just skip the pink one. And then this is a different transcript. This is a different bit of information. Sometimes there are bits of intron that we end up keeping, so we can keep a bit of the black line. There are mutually exclusive exons, so in real data, we observe cases where you get exons 1, 3, and 4, or 1, 2, and 4, but never 2 and 3 together. Yeah.
Variation across genes or across individuals, but on the same gene?
This is variation within an individual. Even within a cell, you can have multiple different transcript types.
corresponding to the same gene, though.
Yep.
You have one gene that produces a bunch of different transcripts.
I see.
Yep. And the transcript diversity of the human genome is not fully known. There are annotation databases that attempt to annotate this as new information comes in, but in working in this area for a while, I can tell you they are woefully incomplete and also contain some transcripts that aren't real, or I suspect are not real. So backing up a step, so this process, the idea that we have an exon here, we have an exon here, and then we have a little bit of sequence that has to be preserved in the intron for the splicing machinery to work. This is actually from a fairly recent paper. This paper was published in 2016. And this is illustrating some of the proteins and some of the components that are involved in this particular splicing reaction. The point of this is not to memorize the names of these proteins. But the point of this is to point out that splicing is a fairly complicated biological process. It's fairly dynamic. And how exactly this happens in different cells is still under study. We know for sure that splicing happens. We can observe the RNA-seq with RNA-seq data and with other technologies. We can observe the sequence. We know that we're missing pieces in the introns and that they're consistently in the same places. But how exactly this happens across different cell types and how it's regulated is not well understood. Cool? Why does it happen?
Why, as in, what's the function?
What's the function? So these mRNA transcripts are going to make protein, which is what I'm going to talk about next. And the sequence of the RNA encodes what the protein's components are and its function. And so in a lot of cases, we can point to damaging variants that lead to intron retention. So if we don't skip these pieces, the protein no longer functions. I think the evolutionary argument for the existence of exons is quite complicated. Okay, so we've gone from DNA, which is identical in every cell, to RNA, which varies widely by cell type, state, and time. It is a single strand of nucleic acids, which is not very stable. And they exist in the nucleus, in the cytoplasm, and beyond. The beyond is more of an exception than a rule. So now we have our translated messenger RNAs in the nucleus, and they get shipped out to the cytoplasm of the cell, where they get translated into protein. So this is very similar to reading the DNA and making RNA, except there's one extra little translation step. And so this cartoon picture, this green gumdrop, is meant to represent a ribosome, which is a small organelle inside the cell. They're quite small. They don't have very many components, but this is a protein structure that's chemically active. It binds to the 3' end of the RNA, and it reads along the RNA, and it reads 3 bases at a time. So a 3-base sequence is known as a codon, and those codons are what give us the information to then build the protein. So I don't know how easy this is to see, but there are 3 little lines in each of these spots. And so there's an amino acid chain, often shown as this beads on a string sort of cartoon. Transfer RNA, remember I mentioned that there were a couple of other kinds of RNA. Transfer RNAs are these little linking molecules that bind with the ribosome and these codons to make sure that the matching is correct. And the key for this kind of code, the way in which this matching happens, is often shown in these kinds of diagrams. So the way you would read this is you read from the middle towards the outside. So your first base in the codon is a G, your second base is a G, your third base is a G, you end up with glycine at that specific codon. Notice a couple of things about this. The first thing is that there's only one start codon, that every amino acid chain starts with methionine, and this is also code for start. So as the RNA is being read through the ribosome, nothing happens until we get our first methionine. And then the amino acid chain starts, and that's GUA. And then we proceed from there using this sort of translation system. Notice that some sequences lead to no change in the amino acid that gets added. So if you have CUU, you end up with leucine. If you have CUC, you still end up with leucine. So you can imagine that this code intrinsically makes the sequence at some bases more important than others, right? Because if I have a change, maybe here, it might not actually matter in the downstream biology, as opposed to something that's the first codon, or the first base inside the codon. Okay, we okay with this? Resounding silence.
Are there?
Yeah.
So I'm trying to just scan the ring. Is
Yeah.
it ever that? Because it seems like they're always grouped based on the third codon, but are there like doubles around that I'm not seeing, or is it if there are like synonymous sorts of things that are always in the third codon? Is that
They're almost always in the third codon, yeah. I think there's some in the second, maybe?
Okay.
This is where it's been a while since biochemistry. I'm not sure.
I just don't see it on in the picture. But.
Yeah. Yeah. I know that it is commonly annotated that bases that are the third base in a codon are often less interesting in annotations. In the back.
I think something that might help me tie together limited understandings of what you do
Sure.
is asking why is the codon not the sort of level of analysis at which we study bioeconomics instead of the base pairs?
That's a great question, and I think the answer is twofold. The first piece is that the codon is not always known, and that in this splicing sort of activity, the codons can shift. So in the splicing, you notice that we have a couple of different versions of this. The exons themselves are not always multiples of 3. So if I go here, I've gone 1, 2, 3, 1, 2, 3, 1, 2, 3. This is actually a great example. And say I've done the first 2 bases, and then I skip to this pink one. I have my third base there. If I go 1, 2, 3, 1, 2, 3, 1, 2, 3, now the third base is different.
So what you mean is that for the same DNA, you could have different codons? Is that
For the same DNA, you could have different messenger RNA, which would give you different codons. That does happen. It's slightly evolutionarily unfavorable. The second, and I think more important reason, is that most of the variation in the genome that we measure isn't actually in the coding region at all. So you could do measurements of codons, but your data set would be much, much smaller, and you'd have much less information to work with. And we'll actually talk about this when we talk about the structure, that very little of the genome actually does this kind of thing. A lot of it does not get translated. And so you'd be losing all of the information that gives us a lot of power over population dynamics, over inheritance, and things that may not necessarily be functional. Okay? Cool. So proteins. Proteins, my supervisor, Ben Neal, likes to say that proteins are like tiny machines, tiny molecular machines running around the cell doing things. And I think that that's a very useful way of thinking about proteins as opposed to these sort of static molecules encoding information. In some ways, you could think of the DNA as the manual and this as the finished product. So protein structure is incredibly complicated. Often we represent this central dogma piece as beads on a string. This is the sequence of amino acids, but these sequences very readily inside of a cell will form three-dimensional structures, and those three-dimensional structures are very widely varied. In general, and this is largely dictated by whether or not the molecules themselves like water or they dislike being around water, so sometimes they will bind to themselves to avoid touching the water, and sometimes they will stretch out. And so you end up with these sort of two gross structures. There's a beta pleated sheet, which is often represented by this sort of kinky line, and then there's an alpha helix, which is another sort of protein formation. And then these can be mixed and matched in many different combinations, and you end up with something that has all kinds of twists and turns in 3 dimensions. And so that's what this sort of squiggly thing is meant to look like. Classifying and understanding the 3-dimensional structure of proteins is very much an active area of research, and it is something that a lot of people spend a lot of time on. This one is hemoglobin, and so you can see that there's actually 4 different protein chains, and they're complexed together. Hemoglobin is the molecule that carries oxygen in your blood. And then there's these little heme groups bound to it. I don't remember what this one is. This is a protein involved in cystic fibrosis. And very often these 3-dimensional structures are critical to the protein's function. And again, not all of them are well understood. So this is another reason why talking about the end product is sometimes more difficult since this is a complicated three-dimensional structure, and DNA is one dimension. It's a sequence with 4 values. You can line it up on one line. This is very hard to quantify. Cool? Okay. So, as I mentioned before, proteins do a lot of things. So enzymes, structural components, antibodies, molecules used in transport and storage, and some hormones are all proteins. These are all things in the body that have active roles in changing things or in responding to a stimulus. I actually wrote that. This is Ben's. Proteins are biological machines with a designated function. And we have a lot of these, and so you can imagine that these machines interact with each other in lots of complicated ways. Okay, so this leaves us with our sort of central dogma summary. The DNA, as we talked about before, is identical in every cell. These two are not. RNA is highly unstable. It gets degraded pretty quickly. So measuring the messenger RNA at a given time in a cell is very time sensitive. These guys go all over the cell. This single-stranded amino acid chain is fairly stable and acts in all kinds of ways in biology. And so this is sort of the central information processing pipeline by which we're talking about DNA and what it encodes. Okay, let's talk about our genome. Oh.
No, I was just going to ask, so you mentioned the measure of the mRNA is
Mhm.
time sensitive. So does that mean that when you store a blood sample or whatever,
Yep. Yeah.
it matters where you store it for the mRNA, which wouldn't matter that much for the DNA?
Yeah, it wouldn't matter at all. A blood sample is a great example. So how this matters in experimental design in psychiatry is very, very central to the way we think about things. So in DNA, you can get a blood sample from a living person, sequence their DNA, do a GWAS, and come up with some sort of biologically based hypotheses. If you take RNA from a blood sample, it matters what they ate that morning. It matters what time they woke up. It matters how old they are. Their gender probably matters, and any number of other things. The presence of stress hormones, all kinds of things matter in this place. And also, if you're studying psychiatry, the blood might not be the place you want to look. And most living people don't sign up for brain biopsies, and so getting relevant tissue. Well, I work on psychiatric disease in expression space, and I can tell you that brain samples in this space are very, very difficult to come by. The same is true of any sort of internal organ that's not easily collectible. Yeah.
Proteins are kind of the active machines that are doing stuff that might drive some of the mechanisms that we care about. And they're coming from RNA, which has this link that I can't quite recall with codons that seem to differ even on kind of similar places along the genome of the same person.
Mm-hmm.
Then to what degree, if I know some individual or some populations base pairs, can I meaningfully annotate for what proteins are involved?
That's a great question.
I'm
No, I think I understand what you're saying. Is if I know this, and I know the collective human knowledge, can I get all the way here and predict what protein we're going to end up with? I think that's widely variable. So some of these things have been very, very well studied, things that are common, things that are highly active in a well-studied disease. But some of these things are largely unknown. So it really depends on what it is that you're looking at. There is a whole academic discipline surrounding going from an RNA sequence to protein and what the three-dimensional structure will look like. That is not a community that I'm a member of, but I do know that it exists and that they do a lot of fancy modeling, and they're still not right all the time. So it's not so much predicting the sequence of the amino acids, which is fairly easy to do. It's predicting the three-dimensional structure and the function. That's much harder. So I'd say results will vary widely. So there are a couple of exceptions to the central dogma that I would be remiss if I didn't tell you about, but a lot of times we sort of ignore these as strange exceptions. So there are long RNAs, long messenger RNAs, that don't actually ever make it to protein. MicroRNAs are sort of the shorter versions of those that are thought to be used in signaling, but again, those processes are pretty poorly understood. And long non-coding RNAs, which are like a full-length transcript, except they never make it to protein, or at least no protein has been observed. So regulation of transcription, that's the process that goes from DNA to RNA, the writing piece. And translation, that's the piece where we go from RNA to protein, depend on a lot of factors. A lot of it looks like this sort of thing, where it's several proteins binding together, and those differ by cell type and by organism, by time point, by many different stimuli. And so as a process, that's poorly understood, that there are some individual cases that are well understood. And then annotations for functional regions of the genome, and this is for human and in animal model organisms, changed a lot. And so a lot of these big databases, there are some things that are quite stable, and then there are some things that you always want to have the newest release because it will have.
What do you mean by annotations of functional regions? Like what is that?
So something like the UCSC genome browser, which I'm going to show a picture of. Something like ENCODE, or not ENCODE, GenCode, something like.
What are they for? I guess I meant, like,
what are they for?
Yeah, like, not examples. But like, why do we care if they change?
Why do we care? Well, if you do a GWAS and you have a hit, you want to know if that hit's near a gene, especially if you work in psychiatry and you want to develop a drug target, or you want to know more about the mechanism that causes your disease. You want to know where the genes start and stop. What is the address of this gene and what is known about its function?
It's basically, for our purposes, the stratified LD score regression annotation matrix, all the ones and zeros saying whether a SNP is in an evolutionarily conserved region or
Nice.
whether it's in an enhancer or whatever. Basically, that thing is constantly being updated.
Yeah. Awesome. And we will talk extensively about annotations, probably more tomorrow than today, but we will talk about them. Okay, so now that we've gone through the central dogma, I think answering this question of what is a gene becomes a little bit more concrete, since I gave you a definition, but we didn't define protein and we didn't talk about how we get there. And so there's this sort of beads on the string mentality. This is the DNA. These are the exons, which are just pieces of code in the DNA. We don't always know which ones they are. This little arrow often indicates a transcription start site. So this is saying this tends to be where the transcript starts. And this little bar can often denote where the transcript ends. And then obviously you can have differential splicing inside of this bit. It does happen. Oh, yeah. What's the function of introns? Unknown, largely. A lot of very simple organisms, like bacteria, don't have them. And as soon as multicellular organisms get a little bit more complicated, you start seeing this splicing behavior and this putting together of components in some genes. And this is all kind of evolutionary theory stuff that's not completely vetted, I think, is that some of the exons end up encoding for certain functional components. And so it's mixing and matching functional components. That's not always true, but some evolutionary biologists believe that that's true. Yeah?
Actually, there's a quantitative geneticist named Michael Lynch, who I believe his theory is basically that introns accumulate, they're harmful actually, and they just accumulate through genetic drift. That means essentially by sheer chance. But once that's happened, it's possible to create functional diversity by alternative splicing, and then evolution sort of just makes the best of a bad lot. It just repurposes it for to like create different versions of a good protein.
Yeah. In the back.
Are these exons and introns themselves collections of base pairs?
Yes.
And how many base pairs?
That's a great question. Most exons are in the 400 to a couple thousand range. That's another great question. Often by sequencing both the DNA and the RNA.
And what would tell you?
So if I sequenced, and we're going to talk about this tomorrow when we talk about technology. So if we say this is the DNA, and then I have an RNA transcript that's much shorter. There's a set of statistical algorithms called aligners, and they take a chunk of sequence and they match it to the reference genome. So if generally how this ends up happening is that, say, this chunk maps here, and this chunk maps over there, and this chunk maps over on the end. There's this sequence in the DNA has been clipped out. And because I've done this matching by segments, and lots of people do this many times over, you can infer that this transcript must have come from this DNA, and this is the intron that got spliced out.
That is the one. The spacing gene.
Yeah, so this would be the intron.
So why does the RNA only contain exons?
Why does the RNA only contain exons? Yeah, it does. And this is sort of that process of like, why do we bother with splicing at all? And James just addressed that there's some evolutionary theory around that, but it's not. I think this is one of those places where thinking about biology and evolution as an elegant sort of designed system can get us into trouble, because this is not elegant, right? This is messy, and this is a process that has evolved over the course of evolution of life on Earth, and trying to think about why do we do that in some kind of designed way, I think can sometimes be more frustrating than it is helpful. Yeah.
Do the alignment algorithms take into account the fact that RNA is variable over time and in different states?
No, they sort of assume that they're only interested in the sample that you're looking at. So if you do it many times, then you have the diversity of the transcripts sort of represented.
Got it. But like reasonably within the same person at two different times in the day, let's say, you could get a different estimate for like...
Yeah, you could. Most folks don't have that much money, but yes. One more. You're fine.
Do people do the same genes among different people differ in their introns and exons? So and sort of relatedly, if I'm the Chinese HIV scientist, is one thing that I might unethically want to do just to like remove these introns and see what happens.
Okay.
So analogously, we're sort of doing something similar to that in GWAS to what the Chinese scientists did. It's very different in some senses, but we're thinking about the difference between people that have this many alleles of this length and this many. But we might also be interested in people that have sort of introns of this length or introns of this type.
Yeah, okay. So two pieces to that. The first bit of your question was, do different people have introns and exons in different places? The vast majority of functional proteins, no. But there are examples where, yeah, we do. There's differential splicing across different people. And so if you do an analysis of differential splicing, comparing one trait, you might get a significant result saying this version of the transcript might be linked to the phenotype that you're trying to study. So if it's really important for life, probably not. Splicing is quite conserved in most things. However, there are some examples where you can see different transcript isoforms in different people. And the second one is, could I just turn this whole thing into protein and see what happens, see what it breaks? You could. It would probably, if it's an important biological gene, you'd probably break something. And there are some disease-associated variances. This is primarily in the rare disease space. This is what one of our colleagues, Burrell, works on. She talks about if you have a mutation in this motif that tells the splicing machinery to bind there, you can get intron retention. And sometimes it leads to some pretty severe diseases if the gene is important. She works on muscle disorders, and so she's been able to demonstrate a couple of cases where if this sequence gets goofed up and the splicing machinery can't bind, the muscle proteins don't work.
So just, because I think I'm getting a little confused.
Yeah.
So tell me if this is wrong. My understanding is that the exons are sort of defined at like a human population level. It's like systematically these are the regions that we tend to find in the transcripts. So it's not about an individual person or a sample. It's about like, you know, where are the regions that we tend to see transcribed at different times. So it's not
Yes.
like one person has different exons than someone else. It's
Yeah,
just
they're not totally backwards.
Mhm.
They tend to be similar, but it is not quite as clean as what you're describing.
But like when we do like exome sequencing, isn't that sequencing the regions that like tend to be exons, or is that?
Yes,
Okay, yeah.
but it's by a pretty liberal definition, and the exome baits generally go a little bit outside, and often include intronic variants as well.
Okay.
So like different exons, I've represented it here as these boxes, but it can get a bit messier where there can be a transcription start site here, and then there's a methionine here, 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 and here, and you will observe 3 different versions of the transcript all starting in different places.
So it's just that the boundaries are a little bit, we have this theoretic idea of like, oh yeah, this is the clean version, but the boundaries are actually kind of fuzzy.
Yes.
But okay.
Yeah,
When we
yeah.
talk about exons, they're not just, you know, we have the simple model in our
Yes.
head, but it's not quite as clean.
Yes. So this is the elegant picture that we often try to think about when you take high school biology, but this elegant picture is actually quite messy when you get down to it. And just to add another layer of mess. Oh, I get to talk about something else first. Okay, so we talked about how there's a start codon and there's a stop codon. Another thing that can get goofed up is if some sequence gets accidentally changed to a stop codon. Now you've truncated your protein, right? And so that would probably be pretty disastrous for the function. So we, humans, have approximately 21,000 coding genes in our genome, but that's still about 1% of all of our bases. And so we were talking about, like, why don't we talk about codons? It's because it's such a small percentage of the genome. And then if you expand your definition of gene to include those things like long non-coding RNAs, you end up with something around 56,000. But the function of a lot of those things is not well known. And then one more added complication. In general, we talk about things being coded on one strand. That's this blue strand. But remember that DNA is double-stranded, and that there are, in fact, gene areas where there's one gene going this way with introns and exons marked, and there's another gene going this way. And we have some of these in our GWAS peaks for schizophrenia, which is extra annoying. This is reasonably rare, but it does happen. Any questions on that? Yes?
So would these two be the same genes, or?
So they're different genes. So if I'm reading off of this strand with this configuration of exons and introns, you can imagine that I would have a very different sequence than if I read from this direction on this strand. And so there are genes that overlap in the genome, which can make some association results very difficult to interpret. I had a very long conversation when I was in graduate school about this, where I asked my supervisor, that doesn't really happen. And he pulled up a region of the genome that he was interested in which that was happening in 2 genes next to each other. So it does happen.
So what does it mean? We have like kind of some of the amino acids from this and some of the amino acids from that in the same cell, or something along these lines?
Well, yeah, because you have this process going on simultaneously in a very large number of genes at the same time. And so transcription of this direction and transcription of this direction will not always be the same. But these would be 2 separate proteins, right? Because they would produce 2 separate long molecules of messenger RNA. they would likely be co-expressed for regulatory reasons that we're going to talk about very soon. Mainly that if this area is transcriptionally active, you'd probably get transcription going both ways. But again, with this idea of a lot of other proteins coming together and influencing the rate at which that happens, they wouldn't necessarily be the same. But yeah.
I want to ask about this.
Go ahead.
Way of doing things twice in the same strand. Can you, I guess, can you unzip it once, or can you unzip it starting from two different locations and kind of having a meaningful sequence that is two different meaningful sequences?
So my understanding, if you're having transcription in a locus like this, is that you have an RNA polymerase that's moving this way, and that's translating or transcribing the blue gene. And then at a separate moment in time, or on the other chromosome, or in a neighboring cell, you might have an RNA polymerase moving this way, copying the yellow gene.
I'm asking about going the same direction. You go the same direction.
Mm-hmm.
Once you start a base to the right, and then something that would look like a stop codon is actually looks like an amino acid, and you keep reading it, and there's
Oh, yeah, like missing a stop. That does happen.
stop, and then it kind of like codes for a totally different thing.
Yep. Yeah, and then you can vastly change. It does happen. You can get a mutation that changes the codon. Yeah, everything happens in biology. This was supposed to be the elegant lecture, you guys. So the overarching sort of idea is that in general, this system works exactly as planned. And a lot of our models assume that this model works exactly as planned. But sometimes it doesn't. And that is absolutely an example of when it doesn't. Go ahead.
What do they think about the effect of non-transcribed genes? What are they doing if not?
Their function is largely unknown. The ones that have been characterized well have been shown to be sort of signaling molecules, signaling to neighboring cells, signaling to other transcription factors.
How do they signal if they're not being?
Often it's the raw RNA ends up as part of one of these big binding complexes. But again, a lot of the chemical details of that are not always known. This is not just not known to me, but like not known to humanity. Okay, so despite this being very complicated and very messy, lots of molecular biologists have put in a lot of work to characterize single genes, so their addresses in the genome, where their exons are, where they think their exons are, where they start and stop, and how many different isoforms there are of all of these. And so this is a great resource if you want to look up a gene, you want to know its address, you want to know how many exons it has, you want to know everything that's known about its function. So I looked up a gene, CACNA1I. This encodes for a calcium channel in neurons. It was one of the schizophrenia GWAS hits, and there's quite a bit of activity on this gene going on in the Stanley Center. There's been several papers on it, so I'm allowed to say that. And so this is a gene on chromosome 22. There's several versions of it, and then you can see which genes neighbor it and which genes overlap it. And so this is, I think, a nice resource for looking up your favorite gene if you want to know where it lives and what it does. There are other annotation databases like GenCode and Ensemble, and these two recently merged, so their information is sympathetic. They may have diverged since, but 2 years ago they were the same. And so this is a project that's been going on for more than 10 years. These are these big aggregation efforts to aggregate everything that's known about a gene, everything that we can say about how it's encoded and what that product does. And so these are nice resources. If you implicate a gene in something, you want to know what it does. These are nice little lookup tables. So if there are no questions about that right now, and I'm happy to talk about this also other times, we're going to talk about our genome and how it's organized. So it's going to change gears just a little bit. So We know most of this information. So we have approximately 3 billion bases of DNA in each cell. And we have to preserve the information that's encoded in there. And so inside the nucleus, the DNA needs to be organized and packaged in a way that conserves space. 3 billion bases of DNA is quite a long string if you have them all together. And it avoids breaks and tangles. Like double-stranded DNA is fairly stable, but it is still just a single molecule. So we talked about how DNA forms a double helix. It wraps around proteins. These are often called histones. And their exclusive function, these proteins, is to organize the DNA. So you keep it from getting tangled. And then the chemical properties of these proteins, the histones, cause it to coil again. So we have this sort of double level of coiling. And so this continuous packaged chromatin, chromatin is the sort of packaged version, is called a chromosome. So it's one piece of DNA, it's one continuous piece, and it's packaged in this way. Most animals have several chromosomes, each containing different information. Humans are no different. And I'm just going to flag this. We're going to come back to it tomorrow. That additional molecules will attach themselves to specifically cytosine, but there are other examples. And this changes how we package things. And so I'm just going to mention this now, and then we're going to talk a lot more about this process tomorrow. But the idea that you can add extra little bits of chemical to your sequence to change how tightly this coiling happens. So a chromosome is a continuous piece of DNA. We talked about that. Bacteria in general have one circular chromosome. So it's a ring. And the chromosome sizes vary pretty greatly depending on the species of bacteria. And bacteria is generally called a haploid, which means they carry one set of their genomic material around. We are quite a bit more complicated. So ploidy, this is a sort of old genetics term. It's the number of sets of chromosomes that you carry around in each one of your cells. So haploid organisms like bacteria, then there are diploid organisms like us. So we have two sets of our chromosomes. We have one set from mom and one set from dad. And then there are organisms with more than two sets. They're mostly plants. Apples are actually triploid. Wheat is hexaploid. Strawberries are octoploid. And the list really goes on. There's quite a lot of plant examples that have very high ploidy. All right, so humans have two sets of chromosomes. And this is called a karyotype, this kind of picture. So we have our autosomes. These are chromosomes that every human has, and we have 22 of those. They are generally ordered by size because these were among the first pictures of the chromosomes that we have. And so you can see that we have 2 copies of 1, 2 copies of 2, and so on. We have 1 pair of sex chromosomes. So females have 2 copies of the X. This is a female karyotype. And males have 1 X and 1 Y. And then, as I mentioned before, there's a small amount of DNA in the mitochondria, and that's a circular bit of DNA, thought to have evolved from a bacteria. So we talked about sex differences. And so we have our genomic information. We have it all organized in each one of our cells, and we need to pass it along. So when an egg is fertilized, it's a single diploid cell. And we have lots of cells in our bodies, and so our cells need to divide. And so we think about this process as mitosis. Again, everyone has probably seen this at some point in their life, though I imagine it's been a while. And so interphase is sort of the blanket catch-all term for the cell existing, doing its business, and not dividing. In general, what starts to happen is that in this phase, towards the end, the DNA replicates itself. So before we can start dividing, we have to copy all of this, and we're going to talk a little bit more about how exactly that happens. And then the packaging of the DNA gets much tighter. And so you end up with these sort of chromatids, where they're They're very tightly packaged. Transcription is pretty much stopped, but you have these very discrete units that we can now move around instead of this massive tangle of DNA. And so in metaphase, they line up along an axis of the cell that's about to split. In anaphase, they start getting pulled apart. And then telophase is basically saying the cell becomes two cells, so it's pinched off. How exactly this process happens, I don't think is super relevant for us. What is relevant, though, is how the DNA makes a copy of itself, since this is the piece that really involves genetics. So this is very similar to transcription. In this idea, we have sort of a zipper, but this time the zipper doesn't just unzip a pocket. It unzips from one end and goes all the way to the other end. So topoisomerase, you do not have to memorize the names of these things, is the molecule that does the initial unzipping. And then the two strands actually replicate slightly differently. There's the leading strand. And so this is fairly straightforward. Just like in translation, we go from the 3' end to the 5' end, matching the bases. So A's with T's, G's with C's. This one is called the lagging strand, and the reason for that is because we still have to transcribe this way, but we're unzipping that way. This is a bit inconvenient, and there's a little bit more molecular machinery involved on that side of things.
Are there ever errors in this process?
There are. In fact, there's a lot of different kinds of errors in this process, which is what we're going to talk about right now. So somatic mutations, when we talk about, this is a term that you might hear in the genetics literature. This means errors in this process. So there was an original cell. All of us existed as a single cell at some point. And there were many, many cell divisions. And this machinery can mismatch bases. So you might get an A in your original sequence, and somehow it gets changed to a C. That does happen. Skipping one or more bases. Adding one or more bases. You can get replication slipping, and this is related to the conversation we were having yesterday during the discussion when we were talking about CNVs, and this idea that you might copy one region more than once, especially if it's not very specific, and you might get mitotic errors where one chromosome just goes to the totally wrong place. So by definition, a somatic mutation is the change in the daughter cell's genome compared to the parent cell. And these do happen, and the vast majority of them are benign, and we never know about them. So these second two, this is what we were talking about yesterday, just that there are two kinds of structural variants that we end up seeing. So it's my cat example. So specific somatic mutations can occur at certain points in development, right? Like we go from being one cell to 2 cells to 4 cells, and we start developing axes of top and bottom and right and left. And so this cat had a somatic mutation at some point during development in the gene that encodes for their fur color. And so this is a black and white, like a tuxedo cat, with half of its face is gray. And this is indicative of a somatic mutation as the cat was developing. So this is an example of the genotype, and the cells on this half of the cat's face would be different from the cells on the other half. Some scarier versions of somatic mutations might include cancer. So it's particularly if we get somatic mutations that mess up the function of the genes that regulate mitosis, they regulate cell reproduction, you can end up with cancer. Because if that gene is broken, mitosis might proceed unchecked. And so somatic mutations are an important part of understanding genetics. They're not really the part that we're all here to talk about, because we're sort of talking about the inherited piece. Okay, so I talked about, types of mutations, and this will come back again when we talk about inherited mutations. So you can have a SNP, so this is a single nucleotide polymorphism, the idea that you had one base here, and we just changed one position. You can have an insertion, where we just read in a couple of extra bases, or you can have a couple of bases get skipped. And so when people talk about indels, they talk about insertions and deletions. It's variants of this type. People talk about SNPs, which is most of what I'm going to talk about. It's variants of this type. And then there are the structural variants. And these tend to be a bit bigger. So a CNV, you can have a small area of the genome, and there is slippage in this machinery, and now we have 2 on 1 copy of our chromosomes. And a very extreme example of this is if the mitotic division gets totally goofed up and 1 chromosome goes to the wrong side, you can end up with something like trisomy. So we had 2 copies of every chromosome in this karyotype except chromosome 13. If this happens in a single cell, the immune system will probably catch it and just kill the cell. And you will never know because it's 1 cell, as long as it's a somatic mutation. So the processes for doing all of this create mistakes. We talked about that. Talked about these mutations. A lot of times, folks talk about mutations as if they're bad and variants as if something that just exists. And so all variants were mutations once, and all mutations are variants. They're more or less synonymous. It's just mutation comes with the connotation of being new and being damaging. All right, so we generally all think about genetics and genomics as something of the heritable variety, so things that are passed down. And so now we're going to talk about meiosis, which is the way in which cells divide for reproduction. So the idea here is that, 1, we have to make haploid cells, right? Because if you get the entire genome from your mother and the entire genome from your father, and that proceeds for many generations, we will have very high ploidy, and all humans are diploid. So the first piece of meiosis is a lot like mitosis. We need to replicate the DNA, and we need to make 2 cells. There's one thing that's different. And this is the recombination, or the crossing over piece about it. So you can imagine we have a chromosome from dad and a chromosome from mom. In meiosis, they end up lining up right next to each other. And we end up getting these crossover events where the chromosomes from dad and the chromosomes from mom exchange information. Like splicing and like transcription factor binding, this process is chemically quite complicated, but the end result is something like this, and it ends up being quite reliable. So, some things about this process that we're required to know if we're going to model this sort of process is, so recombination is this idea that the maternal and paternal chromosomes are shuffled. You don't get the entire copy of chromosome 1 from your grandmother or your grandfather, the one that you got from your mother. Your mother has 2 parents, right? You get a mix of grandma and grandpa, and this is the process by which that happens. This does not change the genetic information, and it doesn't change ploidy. All we're doing is doing some shuffling. So this occurs more frequently in some places than others. These are known as recombination hotspots. And then the recombination rate is approximately 1 event per chromosome, and it's slightly higher in females.
Is it the same with sex chromosomes, or is it not?
The sex chromosomes are weird. So part of the reason it's slightly higher in females is that the X chromosome can recombine with itself, and the X and Y can only recombine in the pseudo-autosomal region, which is just one end of the X and Y chromosomes. So yeah, they do, but only on one side. Okay, so meiosis 2 is where this gets really, really different. So we're going to divide the cells again, but we're not going to replicate the DNA. And this gives us haploid germ cells. So we have this same sort of mitotic procedure, except we haven't replicated the DNA. And so now we get 1 copy of each chromosome in each cell. So if we zoom out, since we looked at this on 2 pieces, you had 1 cell to start with. We replicated the DNA. We crossed over, so the chromosomes are now sharing their information. We made 2 cells, And then we took our recombined cells and sorted them into 4 haploid gametes. It happens a little bit differently for sperm than it does for eggs. Eggs actually end up making only 1 gamete out of this and 4 things that never become eggs. Whereas the spermatogenesis ends up with 4 sperm. Okay. So we talked about somatic mutations and the mistakes that happen in the replication. This replication machinery is very similar, and so you get mistakes here as well. And when a mutation happens in the germline, now it can be passed on to the next generation, and this is a new mutation. These are called de novo mutations. If you see that in a genetics paper, this means that the parent didn't have the variant, but the child clearly inherited it from somewhere. And again, most of these are benign. So again, we can have all the same types that we just talked about. We can have this sort of SNP thing. We can have an indel. We can have CNVs. And we can have, yeah?
I would say that most mutations are benign is because when they're not, the individual is not born.
Especially in the germline. Yeah, I would stand by the statement that most mutations are benign, especially non-structural ones, because they don't fall in coding regions and they haven't disrupted the structure.
But even when it's not de novo, it could be that it happens and then the just killed.
Yes, yeah. And most trisomies are like that. This is trisomy 13, which is compatible with life, and it has a syndrome associated with it. I think the most well-known one is trisomy 21, leads to Down syndrome. But most trisomies are not compatible with life. And so if you get a gamete that has either 1 copy of a chromosome and not its mate, or it has 3 copies of a chromosome, usually the mother will have a spontaneous abortion and probably never know about it so early in development because the fetus is just not viable. The idea that if this person, so this is a pedigree diagram, and we're going to talk about these a little bit more next time about how to read them and exactly what they mean. But say this person has a germline mutation, they can then pass it on to a couple of their offspring, and then their offspring can in turn pass it on to their offspring, and these are how new population variants get born. You can imagine taking this on fast forward through many, many years of evolution. This is a phylogenetic tree of humanity, and so this is the origin of modern humans in Africa, and then we have some divergence as we expand geographically, and then this branch right here is the out of Africa migration, and so you can imagine that these populations get more and more different as this sort of thing, as this sort of thing goes on for hundreds of generations and millions of years. So this kind of thing can go even further. We can get to all the way to speciation using this, and this gets into the evolutionary genetics field and all of those models. And so that was my introduction to molecular biology. If there are any questions about this stuff, we do have a couple minutes, and I can take them now. Yeah?
This may seem like a simple question, but why do we study SNPs instead of indels or deletions or all the other type of variants that you mentioned?
That's a great question, and the answer is that SNPs are easier to measure. And tomorrow I will talk a little bit about the technologies and how they work and why the SNP technology is so easy. Anyone else? Yeah. Yeah. So in a perfect world, the unit of expression would be the transcript. You would count the number of copies of the transcript that you observed in a cell. Our technology is not that good. And so, again, this will be more tied into the technology talk. But it really depends on the technology. So in an array, you're looking at fluorescence level. So this spot is brighter than that spot. That means this cell has more expression than that one. If you're looking at a sequencing technology, so something like RNA-seq, you're going to count the reads that you see from each transcript. So it really depends on what your data are. Anyone else? Resounding silence? Oh, one more.
So I'm interested in splicing. Is there some randomness happening there? And I was wondering if people have exploited the randomness with respect to splicing the same way that we do with microRNA.
I don't think they have. My gut feeling is that our measurements of splicing are not reliable enough for that to produce a meaningful result. But hopefully someday they will be.
Is the randomness in the splicing, there's randomness within an individual
Mhm.
as well, so it would be tricky. Like it's not, if there were systematic differences in splicing across people, then you could maybe do that. But if all of the randomness exists within the person, it's not clear how you would then translate that into characteristics of the individual as a whole.
Okay.
You mentioned gene expression is often mediated by chromatin state.
Mhm. How is the state decided? So that we're going to talk about tomorrow. But the short version, this is the entire field of epigenetics. The idea that there are things on top of the DNA, so just like those little methyl groups that get added to the DNA, and also how tightly the histones are packaged. And that's a dynamic process where it depends on the sequence. It depends on developmental time point, time of day, and so things are constantly being methylated and unmethylated. Chromatin is constantly being packaged and unpackaged. And again, there are specific examples where that's not well understood, but in general, as a global process, it's not well understood. Cool. All right. Well, thank you guys.